Okay, so next up we have Elma Green from the local team here at the University of Melbourne, who's going to present her work on tracking brain states in neural field theory? Yes, neural field theory.
Almost. Why it's challenging to track brain states in neural field theory. Just 1 minute while I get this up and running. There we go. Okay, thank you very much for the introduction, Dean, and for the opportunities to speak. So I'm going to talk about, I guess, some mathematical explanations for some of the challenges we have in predicting epilepsy. And I'll preface this talk by saying I think that the brain is probably going to teach us a lot more about maths in the coming decades than maths is going to teach us about the brain. But given that, I think we all in the room can agree that seizure prediction is difficult. I've put up a screen capture of Klaas's paper, which is almost a decade old. I think we're still on the long and winding road, but we've had some success. So here's another hit for Mark's Neuromista paper. So that we've had more successes than this, but put up one from our group. But as others have acknowledged during this conference, there's still some issues. There's quite a bit of patient variability in success, so we're not quite there. So I'm going to try and explain why the problem is so challenging and why we might have some more success with some patients over others. And I'll just add that we do have a bit of promise in also Mark's work, but with Andrea Varsavsky and the long-range dependency work that says that there should be some sort of slow underlying process that governs when epileptic seizures occur that might, if we had the perfect signal, make them predictable. Okay, so let's look at this. Purely looking at the interface between EEG, which is the predominantly used measurement, and brain activity. So I've popped this one up there just to re-highlight in everyone's minds the scale of the challenge we're trying to do. We have billions of neurons in the brain, and we take, in comparison, just a small amount of measurements, and we're trying to observe what's happening underneath with that. So for the rest of this talk, I'll introduce a very basic model for networked oscillators and show that that model with an EEG-like measurement has very poor observability with some matrix tricks, and give some implications and conclusions for our work in not just seizure prediction, but in general tracking, predicting, detecting brain dynamics for applications in epilepsy. Okay, so I'll start with an introduction to the class of coupled oscillator models and systems in general. They're very rather lovely. They're quite interesting. They have self-organizing behavior. So they're systems that synchronize without, they don't have a conductor or a leader. They all, the synchronization is, say, an emergent property of the networks. So some examples. The first coupled oscillator system that was documented was by Christian Huygens, the inventor of the pendulum clock. So he invented the pendulum clock in 1657, I think, 7 years before he wrote up his results. And so that's actually a screenshot of his actual manuscript. It's quite remarkable that we still have that. So, when he was inventing the pendulum clock in his kitchen, he suspended 2 pendulum clocks from his kitchen table in the Netherlands, and he set them running. And he noticed that after about half an hour, no matter what way he started his clocks, they'd end in perfect antiphase. So he said that he thought the clocks had some sort of sympathy for each other, and that it was probably the imperceptible movements across the common support across his table that were causing them to synchronize, which is quite true. Much later, the mathematics for this came about, and it is the very small some very small coupling between the two systems that allows them to synchronize. So the other nice biological example, apart from the brain, we'll get to that next, is fireflies in North America and Southeast Asia. There are little flies that at night flash, and they kind of flash at random, and then in mating season, without having any commander or without having any conductor, each of the fireflies takes in information, timing information about how the other fireflies in its immediate vicinity are firing, and they all suddenly kind of adjust themselves and flash all together, which I imagine would be very disconcerting if you were walking through the forest and suddenly this huge flash came out of nowhere, not a human thing, just some fireflies. And so our brain tissue seems to, when we record EEG from it, seems to have this synchronous behavior, and as John Terry discussed, it might be some sort of emergent property of the network. So they're interesting systems. So very basic example of just 4 coupled oscillator networks. So each oscillator is very simply modeled as the dynamics of a pendulum clock. So they have a natural frequency, they're connected together with some small couplings, some alpha terms, and we measure some sort of pseudo EEG from them, which is a weighted sum of the activity in each oscillator, so a weighted sum of the pendulum angles. And I've included the other plot to show that this is an all-connected network, so each one of these oscillators is connected to each of the other oscillators. Some maths, I apologize. It's so basic pendulum that a lot of people would have covered in high school physics. And some parameters, just so I guess this is for people who are familiar with these equations, that they see where what I'm talking about, and for everyone else, you can just keep picturing a pendulum. That will work for the rest of the talk. So there's some coupling, shown in red, and what's coupled is the difference between the states, which in this case are the angles of the pendulum, so there's those small differences. And I'm overall looking at observability, which is the ability to take our EEG signal, or our Y, or output, and go back and find out what all the states, or those angles of each of those pendulums, what they're doing. And that's governed by some matrices, A and C. A defines a network, C defines our measurement. And two little assumptions. I do some linearization. I've looked at this in nonlinear systems as well, but for what I'm presenting today, I'll do the linearized version because it's cleaner, nicer to present. And I've gotten rid of the damping, so it's an oscillator with no damping. Because our brain systems, they seem to, well, looking at the data, they seem to operate on the stability boundary. We certainly don't have uncontrolled oscillations that go mightily massive, apart from seizures for a short time and it corrects itself. And our brains don't seem to get damped down. The oscillations don't disappear. That would definitely be pathological. It would be dead. But some other comments on the model. This isn't new. It was used in 1985 by Wright and colleagues, again to model state changes in brain activity measured by EEG. It's a really generic synthetic model. There's no biology. So it's going to tell us nothing about brain function, nothing about epilepsy. But as the thing is always with models, you pick the model to answer the question that you have. And in this case, the question is, if we've got an EEG-like measure, how much about an underlying system, if it was coupled oscillator-like, can we glean out? So we're not overly concerned about the node dynamics of this network. We just want to know how much of the node dynamics how much information we could pull out. So I've also looked at other oscillators, Van der Pol oscillator, Kuramoto oscillator, and equally the Fitzhugh-Nagamo, one with actual biology, and they're all second-order coupled oscillator models, so they could be pretty interchangeable with this analysis. And so where does this fit into the, I guess, most recent research on observability in networks? Stephen talked about this paper before, or rather its sister paper. So this paper by Liu and colleagues is one of the big ones in complex networks, and I've included their plot. And what they basically say is that you can take very large, big, complex networks, and you can work out the subset of nodes that you need to measure from so that you can observe the activity at all the nodes. So the red-coloured nodes with the subset they have to measure from. But as Stephen pointed out, that works for most networks except very symmetrical ones. And unfortunately for us in the brain, we have kind of symmetrical networks. So this is Stephen's own work. And so going from the very big networks down to just small networks, and his motifs with so coupled oscillator networks effectively with 3 nodes. And it shows that the observability, even in these small networks, is not exactly guaranteed. It's highly dependent on how the networks are connected. So what I've been looking at is back to large-scale networks, but each of my nodes are connected to all the other nodes. So in theory, not in practice, but in theory, I should be able to measure from any single node and get the information from all the other nodes. So just to define observability again, generally if you want to track, predict, control the brain, we need, it's dependent on being able to observe the dynamics. And there's a nice measure that says a system will have poor observability if the determinant of what's called an observability matrix is small. So I'm going to have a look for this pendulum coupled oscillator model, find its observability matrix, and analytically show with some van der Monde matrix tricks that it has a very small determinant, so observability is difficult. And the nice thing about this is we can kind of split the analysis into a network model for brain activity and a channel model for our EEG measurement. So kind of like the intrinsic information content of the system of our brains, and then the readout information of the readout channel from the EEG. Okay, so I'm going to do a bit of pointing. I'm afraid I forgot the pointer. So this is the expression for our observability matrix. It's just a giant matrix with C and A terms. The A governs the network, the C is the measurement. And I've added a wiggle on top of my A's, and that just meant I've converted it to a discrete system, so I've got a lot of exponential terms. It just means it's discrete, which is pretty reasonable because our measurement, the EEG, is digitized, it is discrete, it's not analog. And so I can do some transforming to take one set of differential equations and transform them to an equivalent set, but conveniently my equivalent set has this van der Monde matrix structure. So this is the van der Monde matrix, if anyone was curious. It has rows of complex exponentials, and the complex exponentials have the frequency terms, the natural frequencies of all those pendulums. And with some maths, we can separate it out into, so I was discussing this channel model and a network model, because we want to find the determinant of this matrix, and van der Monde matrices make finding determinants very easy. Nice analytical expression. And now I have a ... because Bill Stacey mentioned that very long, eloquent mathematical derivations are not great for cross-disciplinary audiences, and so the very long, eloquent, time-consuming derivation has been left in the appendix of the journal where it probably belongs, and you'll have to believe a little. So I can take this purple expression here and find an expression just based on the size of the network. So n is the number of oscillators. So we have Where's my mouse? There we go. Our network model, which is a very large number, so there's lots of information in there. And then we have a channel model, which is the product of the C's or the weights connecting us to connecting the measurement to each of the states, or each of the oscillators. So our network model is fixed. We can't do anything to mold the brain to make it more observable, but we can tune the measurement. So I'm going to take this expression and just replicate it on the next slide. It's the same thing, just to give me a bit of room. And I'll have to add a constraint that the weights, the EEG weights are, they have to sum to 1, because otherwise we just get pseudo EEG signals that had exceptionally large amplitudes that were too big to fit on computers. So to maximize that product, the largest channel model would have equal weights. So you'd, the EEG will be the sum of the angles of oscillation all measured equally, not some measured you could have bigger ones that were closer to the electrode, for example, by having all the weights the same. And even with that maximized, doing the best EEG measurement we could, when you pull all this together, we get this expression here, which is a really, really small number. Even for small networks, even if you had 10 oscillators, 5 oscillators, this is a really, really small number. And so if the determinant of the observability matrix is small, it means that these coupled oscillator networks are pretty poorly observable, even when the EEG signal, in a way, has been optimally acquired. So what does all this mean? It's a really abstracted model of the brain network, and yet observability is found to be really poor. It's so abstracted from the real-world problem that we have no noise. All the parameters are known. I know the model, and it's a stationary environment. It's mathematically lovely, and yet it's very challenging to observe. You can also show that it's a theoretically observable system with a Popov-Belovich-Hautus test, if anyone's, if that means anything to people in the audience. And yet practically observability seems to be ill-posed. Let's show one more result. Kalman filters, because they're good at applying to data, and engineers and mathematicians like applying them. So Bjorn showed some Kalman estimates, and if you recall, he had the red dots that were the estimates, and then he had blue lines for the error on the estimate, or the variance on the estimate. So what this is showing here is the error on our estimate plotted against the number of oscillators. So, yeah. So down here, the error, the variance on the estimate is almost 0. It's really low. For small numbers of oscillators, this is 10 oscillators, this is 20 oscillators. So for small numbers of oscillators, the variances of our estimate is really low, which means we can estimate what all those pendulums are doing really, really well. But unfortunately, this has sort of sigmoidal behavior, and when we get to 100 oscillators or even 1,000 oscillators in our network, our variance is 1, and it's all kind of normalized. And so what that means is we're trying to estimate signals with amplitude of 1, and the variance on our estimate is about 1. So the error on our estimate is the same as the thing we're trying to estimate, so our estimate might as well be random. So there's a lot of parameters in the way you can set up your Kalman filter, from the amount of noise you expect on your measurement to the weights, the measurement, how the C matrix is connected to the states. But in general, the sigmoidal relationship seems to always apply. So very quickly, you lose the ability to observe. And when the overall average variance is high, that doesn't mean we can't observe anything. It just means that in a network of 1,000 clocks, we can still only observe 10 to 20 of those oscillators. And also, so numbers of samples is another variable. I've increased the amount of samples, or the amount of time I've allowed the Kalman filter to work out estimates, about 100-fold between the black line and the light grey line, and there's not much difference there. It didn't do very much better. So what's the implications and conclusions for all of this? I would say that large-scale observability is pretty utopian for large-scale oscillatory biological systems, which means that prediction success starts to become highly dependent on the precise location of the electrodes. And of course, we don't know exactly precisely where to put the electrodes. And I just referenced one of Greg Worrell's papers from 2010, which had co-recorded macroelectrodes and little microelectrodes next to it, and was able to show that there was seizure activity on the microelectrodes that weren't being seen on the larger electrodes, or indeed were seen much later when the seizure activity spread. And I think that's kind of lends itself well to this analysis when you can only see a small subset of the actual oscillators that are there, only the most spectrally distinct, the ones with the angles that are farthest apart or the frequencies that are farthest apart, and largest amplitude are the ones we're going to see on our electrodes. So where we might think we have an electrode and it's covering a certain area of the brain, it's only covering the most dominant information there, and I don't think any signal analysis on that signal is going to pull out what basically falls under our noise floor. So the high variability in patient performance that we saw with the NeuroVista trial is probably to be expected. The same patients might have had better performance if the electrodes were moved slightly. So some interesting challenges for us in this community to get sufficient information out for therapeutic benefit in epilepsy. So we can concentrate on tracking, say, local dynamics in small subnetworks. But it's not exactly clear if really understanding a small local network can tell us what the emergent behavior of the entire network might be. And so the other thing that Karen was also discussing earlier is active or evoked EEG or probing. And so Kalupson and Mark Richardson also doing it. Dean has been leading the probing here in Melbourne. It's very exciting. It has a bit more, I guess, theoretical promise, because if we're putting signals in, we can maybe influence which of those small subset of oscillators we can push over our measurement threshold so that we can see them. But it makes the big open question, what should the probing signal be, or how do we design that probing signal? And perhaps Lizette's comments from earlier in the week, where we probably need to know a lot more about the mechanisms to be able to design a probing signal that works really well. So to conclude, and concluding comments before questions. So I guess this work, it shouldn't be anything that's of any surprise in the community. It's hard to do seizure prediction. It's hard to get information out of the brain. But perhaps this is a bit of theoretical justification for why it's so hard, and also a theoretical push that maybe we need to be probing to kind of force more information out of the system. Some paper references that contain the work was talking about here, and big thanks to our Melbourne Epilepsy group, both at the School of Engineering and also St. Vincent's Hospital, for all the most interesting and insightful and informative discussions over the past years. Any questions?
Observability of the EEG from a patient with the predictability success that you have in that particular patient, so that you can tell you whether the patient will be seizures or the dynamic activity from the patient will be prevalent or not, based on how observable could be. Because I mean, not all patients are good candidates for. I mean, we have pharmacoresistant, we have patients which are not candidates for surgery. So why not to accept that it is not our failure to predict a seizure, or that we are not sticking the electrodes in the correct place, or that we are not sticking the good electrodes, but just that the system is not predictable.
Yes.
And therefore, trying to predict something there is impossible.
Yeah, it could quite possibly be that it is quite predictable. Oh, sorry, quite unpredictable. But even if it's, I guess, even in the case that you make the assumptions that it's predictable, you use the long-range dependency work Mark talked about to say, we think there's some underlying slow process in there that could be predictable. Even then, this work is saying in that case, it's incredibly hard to get the information out. So I agree. If it's not predictable, then we have a big problem.
I mean, that's the point. The point is, why not? I mean, we have been discussing in this community whether we can predict or not predict seizures, and then we try to see whether the mechanism will tell you more information, etc. So maybe after those years, we have come to a point to accept that we have done the best in our hands, and based in all these sophisticated mathematics, it can be. Because there are some brains that will not be predictable. So I'm sorry you have an unpredictable brain.
Yes.
And therefore, you will not be a candidate for seizure predictions. And at the end, I mean, why not accepting that we have found a new solution for some patients, but not for others?
Yes, I mean, before say focus sensitive epilepsy, the best predictor will be somewhere that way, nowhere near the brain. But the other one is so the definition so the observability also applies to tracking, monitoring. So early monitoring, if we can detect early enough. So if we can detect when a person that doesn't have predictable seizure, but if we can detect them early enough, that could be sufficient for therapeutic intervention. And so the observability would apply just the same. If it's a very unobservable system, it will be very hard to observe when the seizure is starting. If that is right.
Okay, we're going to go to Steve, and then up here, and then up here.
Okay, so just a gorgeous story. But I have a suggestion. I think that in general, neurons and nuclei in the brain are all directed. They don't have bidirectional couplings. That's number one. And so if you show all-to-all bidirectional coupling, I would have been shocked if that was observable. The second is that even if there's enough graph-directed coupling to prevent observability, you can prune it back to what should be underlying observable pieces that expand that network. And so, have you thought about converting from bidirectional to unidirectional couplings, and then pruning back connections to see if you can indeed create what would be potentially a very large number, but finite, of observable.
But yes, I haven't, but I am now. That is very interesting to see in that case where we get a lot more than the very minimum number there. But what's the limit of it still?
And then it goes back to the sets question, because you know a lot about the connectivity. So you could inform the network based on what we know about the physiological connection. That would, shall we say, bootstrap you very far along to then microclap the topology of the line.
And this is individual. I mean, and that's why in some cases, for some patients, it works and the other. And it's wired. It's not magic. It's just your brain behaves like that because it's wired as well.
I got a question from the back.
Yes. Well, you're treating the brain as a sort of an isolated thing. So obviously we can predict human behavior, and that's a product of what's in your head. And that's because we understand how people react to inputs.
Yes.
And you're able ignoring that
I'm
completely.
ignoring.
So, I mean, we can, you know, this, I think this suggests, if your theoretical work is correct, that we need to take into account learning reactions so that we can predict better.
Yes.
And seizures are resulting from a process. And if we measure every input, in many, many cases, we should predict the occurrence of seizures. It's just that you're treating the brain as a cell.
Yeah, I agree. When you pretend that we're using a bubble, then yes, it's very unobservable. And not only can we put in probing inputs to try and find out what the system's doing, we can measure the inputs that are happening anyway.
Okay, I'm just going to have the last question up here.
Yeah, so very nice work. One question or concern, I guess, is in your computation of observability, you're trying to observe every single thing, right? And on seizure prediction, you don't need to know every single node in the network, or you don't know exactly what we know, but we really only need to know one-dimensional thing of the likelihood of seizure. How many observables that takes, and what dimensionality that most people don't know yet. But I think it'd be really nice to have results describing the observability of not everything, but some subset you have to.
I guess the thing was that for me was the disparity was, how much information, how big the system was, and how little you would get. And by just passively acquiring the EEG, you don't get to steer or direct what information you get. You just have to hope. Well, the information hasn't been in there for what we found. So, I guess, yeah, we should be exposing to try and steer this sort of information.
There's a lot we don't maybe it was enough.
All right, let's thank Elma again. Okay, so